Hello, my name is Daan and I'm going to tell you about the molecular toxicology of phthalates, specifically DEHP. Let's start with what are phthalates. Phthalates are common plasticizers used in vinyl flooring, medical devices, PVC, and food packaging. And as you can see on the slide, there are many types of phthalates that are going around. Now we are going to talk about DEHP. What is DEHP? The full chemical name is bis-2-ethylhexyl phthalate, and it is used as a plasticizer to make PVC more flexible and more durable. It is the most extensively used phthalate around. And unlike other persistent chemicals like PFAS or dioxins, DEHP is rapidly metabolized into mono-2-ethylhexyl phthalate, in short MEHP. The problem with DEHP is the constant exposure, which leads to a continuous eternal dosing. The ATME processes absorption and distribution, and its effects on DEHP. DEHP is a small molecule and highly lipophilic, so it easily passes passively through the gut. As soon as it is absorbed, it is metabolized into MEHP, and the distribution is done in the form of MEHP. The metabolism. DEHP is considered just an environmental factor. It has a low acute toxicity and is an inactive parent compound. However, this all changes when it is hydrolyzed into MEHP by carboxyl esterases, CES for short. The role of MEHP is different. It is a biologically active toxicant. It has a high endocrinic disruption and it suppresses the steroidogenesis genes. The main target receptor is the PPAR alpha receptor, and that goes for both fetal toxicity and in adults. The elimination. As you can see in the graph on the screen, DEHP is rapidly metabolized into MEHP within 5 hours of ingestion. Both compounds have a short half-life and are typically eliminated through the stool or urine within 48 hours. The levels of DEHP found in urine are neglectable. In order to understand why DEHP is so concerning, we first have to look at the organ it targets the most, the testes. The testes are a unique dual-function organ. They have an exocrine role, which is the production of sperm through spermatogenesis, and an endocrine role, responsible for for synthesizing testosterone and other androgens. The exocrinic and endocrinic functions of the testes are physically separated into different compartments. Inside the seminiferous tubules, we find the Sertoli cells, which are often called the nurse cells, because they provide the structural and metabolic support for the developing sperm. Just outside of these tubules, in the interstitial space, reside the Leydig cells, which are the factories for male hormones. MEHP suppresses the genes involved in steroidogenesis in Leydig cells, and this most likely occurs through the PPAR alpha receptor. It is currently unknown by which means MEHP interferes with the Sertoli cells. Currently, it is still unknown where the exact interference takes place within the Leydig cells. However, it has been narrowed down to an interference within the pathway shown on the left side of this dia. While we have discussed how DEHP targets the testes, the clinical And morphological result of this interference is a specific cluster of developmental disruptions known as Phthalate syndrome. This term describes the collective damage done to the male reproductive system when exposed to these chemicals during critical life stages. At its core, Phthalate syndrome is characterized by four primary internal disruptions. First, a significant reduction in the testosterone development. Second, the impairment of both Sertoli and Leydig cell function. Third, a visible alteration of the testicular architecture. And last, the induction of apoptosis, or programmed cell death, in germ cells. Key symptoms of Phthalate syndrome are a reduced anogenital distance, cryptorchidism, undescended testis, hypospadias, and a reduction in both genital size and sperm quality. While DEHP exposure is a concern at any age, the level of risk is not uniform across a lifespan. Research has identified a masculinization programming window, which is a narrow, sensitive period during the first trimester of pregnancy that is absolutely vital for the male reproductive development. During this specific gestational interval, fetal Leydig cells must produce adequate levels of testosterone and INSL3. These two hormones are responsible for the biological signals that drive the differentiation of the Wolffian duct in the epididymis and vas deferens, the formation of external genitalia, and the proper descent of the testis. If this hormonal signaling is suppressed by MEHP, even transiently, during this window, the developmental trajectory of the fetus can permanently shift. This brings us to the concept of developmental risk. In toxicology, we distinguish between organizational and activational effects. Exposure in adulthood typically results in reversible functional changes. For example, a temporary drop in sperm quality might recover once exposure stops. However, fetal exposure causes organizational effects, meaning it permanently shapes the anatomical structures of the body. When DEHP interferes with the critical window, it can lead to lasting structural defects that cannot be undone later in life. Beyond the timing of the development, there is a toxicokinetic risk. Fetuses and neonates have an immature metabolic and detoxification system. Their hepatic enzymes and renal clearance mechanisms are not yet fully developed, meaning their bodies cannot flush out bioactive metabolites like MEHP as efficiently as a result. Adult. To truly understand the molecular impact of DEHP, we have to look beyond individual hormones and examine the transcriptome, the complete set of gene expressions within a cell. Recent transcriptomic research provides a bird's eye view of how DEHP exposure rewires cellular regulatory networks. When a cell is exposed to DEHP, we don't just see a single change. We see a coordinated response involving multiple high-level signaling pathways. Specifically, DEHP significantly activates inflammatory, growth factor, and the stress-associated signaling. This includes the activation of the MAP key-related cascades and pro-inflammatory cytokine signalings like the IL-6, JAK, STAT, and IL-17 pathways. This tells us that the cell isn't just reacting to a hormone mimic, it is entering a state of high cellular stress. In conclusion, first, the exposure problem. While DEHP is rapidly metabolized and excreted, the environmental presence ensures for a continuous internal dosing. Second, the bioactive threat. While DEHP is an inert factor, the primary metabolite MEHP is an active toxicant, which can initiate endocrine disruption via the PPAR alpha receptor. Third, molecular disruption. MEHP chemically suppresses the steroidogenesis pathway in the Leydig cells, thereby severely inhibiting the testosterone biosynthesis. And last, developmental permanence. While adult exposure causes reversible functional changes, fetal exposure can lead to a permanent and irreversible structural defect. This is the end of my presentation on phthalates. I hope you have learned something from it. This last slide shows an overview of what happens after DEHP exposure. The averages in the right bottom corner are based on measurements taken in the Netherlands. Have a nice day.